Welcome to the Mucosal Immunology podcast, brought to you by the Society for Mucosal Immunology and Mucosal Immunology Journal. Each episode will feature in-depth interviews, discussions on the latest scientific research, and real-world applications that are transforming healthcare today. Available on all major podcast platforms.
Welcome to another episode of Mysteries of Medical Histories. Hey Ashu, how are you?
Hey Mayan, I'm good. Wow, I cannot believe that we are already in our second episode. It has been a month already. That remind me, do you remember last episode's puzzle?
Ooh, Ashu, I'm afraid I don't. It's all a blur now. But what I know is that we had a great response from our listeners. They were all over it trying to crack the case.
Classic Megan. Always keeping things fresh. All right. How about we pick a lucky winner for last month's puzzle? The mystery on the street from all the guesses we received.
Ooh, I'm excited. All right, here we go. Well, actually, our special guest, SMI member Dr. Catherine Knoop from the Mayo Clinic in Rochester, will announce the winner. Drum roll, please.
Congratulations to Samantha Jensen, an SMI trainee member at Weill Cornell Medical College in New York, for guessing correctly. Thanks for listening, Samantha.
That's awesome. Congratulations. It seems like our listeners really love a good mystery. Who does not enjoy solving a puzzle, right? So for all the listeners, the answer to last week's puzzle, the disease was cholera caused by contaminated water, and the mystery hero was John Snow, who is also considered father of epidemiology.
John Snow. Now, let's get into today's puzzle. If you have an answer, please email it to mucimmunol@gmail.com. That's mucimmunol@gmail.com with the subject line podcast clue. So this one is pretty spooky, Ashu. Listeners, buckle up because we're heading back to late 18th century. We're deep in the French Alps and something sinister is happening in a small isolated village.
Well, imagine living in a time before antibiotics, even before vaccines. A single cough could mean the end. And in this case, it is a sickness that seems unstoppable. It starts with a fever and raking cough, then come the seizures, the delirium, and finally the silence.
Oof, the villagers are desperate. I would be too. Their usual remedies are useless against this unseen enemy. But amidst the despair, we have three individuals that remain, each with a different approach to survival.
We have the village healer. Her knowledge of traditional medicine passed down through generations. Will her potion and tinctures be enough?
Then there is the blacksmith, a man of strength and resilience. But even the mightiest oak can fall to a relentless storm.
And finally, there is this mysterious stranger. He seems to be keeping his distance from everyone else, almost like he knows something they don't.
So one of these three will survive this nameless plague. But who, Ashu? Who will survive? What do you think?
I'm not sure, Marianne, but I'm sure our listener will know. All right, listeners, now it is your turn to solve the puzzle. Who survives among these three? Email us your answer at mucimmuno@gmail.com. That is mucimmunol@gmail.com with the subject line podcast clue. And the first one to get it right will get a shout out in the next episode.
See you next episode. Now for our Mucosal Research Roundup. We have Catherine Knoop here to share the latest at Mucosal Immunology. Hi, Catherine.
Hi, Marian. Today, let's talk about the Emerging Leader Collection at Mucosal Immunology. This is a special collection of literature reviews from rising stars in the field, covering topics on all the barrier and mucosal surfaces. First up is Eduardo Villablanca from Karolinska Institute in Stockholm, Sweden. His review combines game theory with host microbiota dynamics and is titled Organismal Mucosal Immunology, a perspective through the eyes of game theory.
How fun! You know we'll be interviewing Eduardo on this very review soon, so be sure to read it. Then come back to check out our longer conversation with Edu in a future episode.
I can't wait for that discussion. Have you heard of the prisoner's dilemma?
It rings a bell.
So it's this classic thought experiment describing how individuals can cooperate or defect from the larger community. So applying this to how the host and microbiota must cooperate is a really fascinating exploration.
Wow, thought-provoking.
Next is Xu Wang at the Chinese Academy of Sciences in Beijing, China. Her review is titled Immune Crosstalk Between Respiratory and Intestinal Mucosal Tissues in Respiratory Infections.
Who knew your gut could be so invested in your breathing, right? This review describes the intricate immune crosstalk between the lungs and the intestines and proves that what happens in the lung doesn't always stay in the lung. It's a mucosal immunity plot twist. Be sure to check it out.
I know, crosstalk is fascinating. Next up, Melody Zhang from Weill Cornell School of Medicine in New York in the US writes about lifelong partnership gut microbiota immune cell interactions from infancy to old age.
You know, I've been a big fan of Melody Zhang's work on breast milk IgG, so it's really exciting to see her zooming out to the full lifespan. This review is like an immune time machine. It describes how our gut microbiota trains, maintains, and, let's be honest, sometimes betrays our immune system, from birth to old age.
You know, I'm also a fan of early life immunology, and this review is all-encompassing. Finally, Matthew Hepworth at the University of Manchester in the United Kingdom crafted a review titled There's No Place Like Home, How Local Tissue Microenvironment Shapes the Function of Innate Lymphoid Cells.
Yes, ILCs. They may be rare, but they really pack a punch when it comes to immune regulation. This review highlights how these cells aren't just immune responders, but they shape their local environment in ways we're only beginning to appreciate. Well, thank you, Catherine. This is a great group of reviews to read.
Absolutely. You can read all this and more at mucosalimmunology.org, home of the Mucosal Immunology Journal, the SMI-sponsored journal dedicated to all things mucosal. You can find reviews, primary research, and research collections from our entire archive, all available open access. Are you ready to submit your research to Mucosal Immunology? Your format-free initial submission will be given a first decision by an editor in less than 1 week. If you have a shorter story to tell, try our brief reports with up to 4 figures and a combined results and discussion section. Submit today on editorialmanager.com/MUCIMM.
Grab your lab coat or your driving wheel or a cup of coffee. I am Marion Brunk, and with me is Agnes Garcia, your host on the Mucosal Immunology podcast. Hi, we are here with Dr. Cecila Muglia, and she is working at Instituto de Estudios Inmunológicos y Fisiopatológicos del CONICET at Universidad Nacional de La Plata in La Plata, Argentina. Good morning, Cecilia. How are you?
Good morning. I'm very happy to be here and to be able to share my studies from science in Argentina.
Hi, Cecilia.
Hi,
super nice to
hi.
have you. So to start with this little interview, could you introduce yourself as a scientist and tell us a little bit about your main research topic in your lab and what techniques and models you use?
Yes, well, I'm a biochemist, and in our lab we study mostly mucosal inflammation, especially in the gut. So we have some topics related to IBD and some related to food allergy. And with food allergy, we collaborate with the Children's Hospital of La Plata, and there's a side of the lab that works with mouse models, but I work mostly with human samples from biopsies or surgeries. So we also we do a variety of things. We work with T cells. We're making organoids from gut tissue. We're searching for ALC, and so we have this wide spectrum of things we're studying. The main investigator of my lab is Dr. Guillermo Docena. I want to mention him. So, and I'm working here from like 14 years now, and I'm very happy. It's a very nice place to work in.
Great. I would like to talk to you specifically about your most recent paper in the Journal of Leukocyte Biology. It is titled Food-Sensitized Pediatric Patients Show Colonic Cow's Milk Protein-Specific Th2 Cells, and your corresponding author on this work. So you work with pediatric patients in this work, and I think it's, as you mentioned on various occasions in your paper, it's very difficult to do that. I was wondering, these patients were referred to the study because they had rectal bleeding. At the time of recruitment, did you know they were allergic to cow milk's protein?
No. The thing is, a few years ago, the doctors from the gastroenterology unit at the Children's Hospital called us and told us that they have found that most kids with colonic polyps had high IgE blood levels. So they wanted us to study these polyps to search if there was any relation between the polyps and, they suspected cow's milk allergy because it's very common here in Argentina. So they're the ones who decide which children to study. If they have rectal bleeding, they do a colonoscopy, and if they have polyps, they send them to the allergy unit. And we find most children have high blood. I mean, we corroborated with what they have found the primary finding that they did have high IgE levels, and we started to study these polyps. The polyps are resected because it's the way to stop the bleeding. So part of it is sent to pathological anatomy, and they give us the other part of the polyp. And we also get a biopsy from the surrounding tissue and the blood sample. So we have, obviously, we had to approve all the protocols in the hospital. Mostly the difficulties were from having the biopsy from the surrounding tissue. But after speaking to the committee, the doctors, anthropologists, sociologists of all different people of all different backgrounds, they approved the work.
Okay, thank you. And those polyps, their formation is related more to inflammation, or this is linked to the allergy process in the gut?
We think they are linked to the allergy process. These kids don't have the typical milk allergy in a sense that they are usually older than 2 years old. So it's not the baby that tries the cow milk and starts vomiting or has serious gastroenterological problems. They just have these polyps, and sometimes they have a lymphocyte infiltrate in the gut. And in the colon, no? It's a rectal or colon. And when we study the polyps, we find they are high IgE levels. We found Th2 cytokines. We found different high proportion of eosinophils. So that made us all think of an allergic origin. And that's when we also studied specific IgE for food allergens in the bloodstream and in the polyps. And we found specific IgE for cow milk protein or for peanut or for egg. So it's probably not specific for cow milk, but related to some food allergy or sensitization, because not all of the kids have other manifestations. Some of them have dermatitis, some have asthma, some have chronic rhinitis, but some of them don't have anything else. So we can't specifically speak about allergy because we can't do a typical control here of a double-blind placebo and the challenge with the antigen, since the response is like late. So in order to better characterize this polyps, we decided to study the T lymphocytes that are present there. So we did T cell lines and T cell cultures specific to cow milk proteins, and from the peripheral blood and from the polyps. And we found that we do have specific T cells for these antigens, and these cells are secrete IL-4, IL-13, and IL-5, and have a typical Th2 pattern.
That's quite interesting, Cecilia. I actually wonder, talking about the allergy, so does an early life allergic reaction to cow's milk protein increase the risk of developing additional food allergies? And
yes,
if that happens, because as you say, you study these polyps from patients, like juvenile patients, right? So they are older than 2 years old. So if this is the case, that they have increased risk for more additional food allergies, if you know the immune mechanism behind it, and is it something that in the samples you have, you would already see that? Have you studied that too?
Yes, in some of the samples we see specific IgE for more than one allergen. I mean, cow milk and peanut, cow milk and egg or soybean is typical. The cross reaction with soybean proteins. So here in Argentina, we don't have the typical. It's the common peanut allergy that you see in the rest of the developed world with anaphylaxis is quite rare here. It's more cow milk allergy is more common. So yes, and this case usually increase their reactions towards other allergens. But of course, we don't have a follow-up study on them. It's mostly from what the doctors tell us, because they see them throughout childhood, and it's most from the information we get from them. We haven't done a follow-up of patients.
So I wanted to ask about the specific T cell responses that you found in your paper. You created some clones, right? You collected T cells from the polyps and from peripheral blood, and then you stimulated this with cow milk proteins, and you created those cow milk protein specific T cells. But then I think you found that those clones that came from peripheral blood T cells had no response to the additional proteins that you tested. Is that correct?
No, we found a similar response from peripheral blood compared to clones from the lamina propria of polyps. So we didn't find specific responses. What we did was to study cow milk's antigens. I mean, alpha and beta casein, lactoglobulin, the main allergens, and we didn't find major differences between clones from both origins, which we really don't know where the allergic, the immune reaction is occurring, where is it occurring in the small bowel? We have no access to the small bowel of these patients, or whether it's just what we see in the colon. So finding similar specificities make us think, well, many of these clones probably are coming from the colon. But we have to do further studies, of course, in order to be able to say this.
Thinking about your research topics in your lab, and you focus on both on allergy, food allergy, and IBD. And I'm thinking with IBD, we know it's a multifactorial disease, right? And one of the key components, apart from genetic background, also is microbiota. And I wonder if the cow's milk protein allergy, to be more specific on that, can also be sensitive to the microbiota. If it is something seen that some kids can develop it because of the microbiota in early life, it's different or altered. And question number 2, is there also a genetic background that can influence being more prone to develop food allergy?
Well, about what you're saying about microbiota, there are several articles, especially Catherine Nagel, that describe dysbiosis in patients with food allergy. We have done studies in feces from healthy children and from the patients with polyps, and we find differences. We're finishing a manuscript with this data, and we also studied the microbiota in the polyps. And so we find like an allergy signature in feces, and what could be a more like inflammatory signature in polyps. And also we find some bacteria. We've studied a few polyps, it's like 9. So it's hard to get statistically significant results. But it's also very hard to get feces from children. They don't like giving feces, and also polyps. What we find is an inflammatory signatures and some bacteria that are found in adenomas and in colorectal cancer. So we think this is an interesting thing to study, because there's not much information on whether these kids have a higher risk of developing adenomas and colorectal cancer as adults. And there's only one paper that says that some of these polyps in older children, like 18 years old, can have fragments or segments with adenoma-like. We don't find that with our cohort, but they are very young children, like from 2 to 6, 7 years old. So we don't know what will happen to them later on. And that's why we're interested in these microbiota studies.
Going back to one of your results, you looked at CCR9, which is a gut homing receptor, and CCL27, which is its ligand. And so you identified those cow milk protein specific T cells that were CCR9 positive, but you also identified the CCR9 negative population that also responded to cow milk protein. Could you tell us a little bit more about this CCR9 negative T cell population?
We didn't study them further, but some of those patients have other manifestations like skin. There was one with dermatitis, there was one with urticaria, and they usually have some respiratory manifestations. So maybe these lymphocytes are headed towards those places, and they are responsive to milk. So, rhinitis is quite associated with cow milk protein allergy. So maybe they're the ones headed to those places. Again, our question is whether they are being formed, in the small gut, and they're traveling to the polyps, or whether they're being differentiated in the polyps in the colon and travel elsewhere. Polyps are small, so it's hard to think that all the IgE that's going around and all the T responsive cells responsive to coming from those small polyps. So probably there are other places where sensitization is taking place, except that we have no access to those tissues.
It would be great to be able to access to different types of tissues, so then you have a more integrative and systematic response and be able to analyze it, right? So
yes, but actually, I don't think the kids' parents will be happy.
Of course not, right? Yeah, it's quite difficult already working with human samples, and I guess even more with pediatric samples, right? So actually, going back to what you say, that some of these patients have, I don't know if you call it comorbidities or other symptomatologies, right? I've been thinking that, or like based on the paper, you say that this gives light on maybe a T-cell targeted immunotherapy for these allergies could be something to be considered for therapeutic matters. And I'm thinking that in early life, the Th2 response is quite important because it's also when kids are growing. And the Th2 has also this regenerative and tissue modeling, right? They are growing. So it's sort of important. We see that also with in the lung with asthma, that they have maybe a viral infection and then they develop asthma because of this prone Th2 response that is actually important for the body, right? So what would be the idea? Very open question, and just wondering what would be the idea of this, how to target this CD4 Th2 response to actually be specific to avoid the allergy, but keep the important arm of this Th2 response.
Well, really, if the only symptom the kids have is the polyps, maybe it's unnecessary to go through a therapy, because they take out the polyps and the problem is like gone. I think the best, I believe we here in the lab, we work with mice and immunotherapies and yes, and the IgE monoclonal antibodies. I think that would be better, because it's not such a serious problem like IBD to go for like an alpha-4 beta-7 antibody, anti-antibody. I think that will make more noise than what it would solve. Yeah. That's maybe one of the reasons these polyps haven't been studied so much around the world, because they're not so serious for kids. But we're worried of what will happen to those kids in the future.
So yes, you're talking about the allergic microenvironment in juvenile polyps. How does that compare from the literature to adult polyps? Is that something that has been studied? And I understand that from a DOHAD perspective, you're interested in young humans, but how does that compare to adults?
Well, information from adult polyps is mostly from adenomas and serrated polyps, so it's quite different. There you don't have for example, IgE synthesis like you have in these tissues, And so they're not easily comparable. You don't find, they're quite different. These are what they call harmatose polyps, also in theory of inflammatory origin. They're quite different. You don't have one of the things they first did when they started studying these polyps in the hospital was to study the different mutations usually found in adenomas, and you don't have those mutations here. Like
okay, so the etiology of those polyps is quite different.
Probably, yes. Like you don't have mutations in catenins or in the APC or in the typical pathways for adenoma, carcinoma, colorectal carcinoma pathway.
Is it the first report, from the best of your knowledge, of IgE secreting polyps?
Yes, and also it's the first report from our knowledge of IgE synthesis there in polyps. We were very happy when we obtained those results where we could prove that IgE was being synthesized in germinal centers in the polyps by a direct and indirect class switch. And that we want to go on studying this IgE to see, whether they are of high or low affinity. And so we still have a lot of studies ahead.
I actually have a related question to that, because it's also what we talked a bit before. So first of all, will you follow these patients in a longitudinal study? Is that the plan?
Yes, we plan to follow them. It's not very easy because it's a public hospital. So if patients don't have any manifestations, they don't come back. We do have some children that go back because they record they have polyps every 2 years, every year. So they come back. So we do have information on some of these children, but since the hospital attends children till 18 years old, we're up to there. Yes. We have a very nice story regarding one child who had a huge polyp, a very big polyp, 5 centimeters, and it was taken out. And the doctors, because they just didn't know what to do about it, they told him they gave him a milk-restricted diet. So he stopped having polyps and manifestations until 2 years later, they said, well, you're fine, you can go back drinking milk. And yes, and 6 months later, he came back with a new polyp. So we can't demonstrate anything, but it was a very interesting finding.
Very much. So my question on that was more because seeing that this is one of the first study showing the IgG secretion, especially in the gut, right? And how there's research in polyps in adults, but they are very different because they come from different type of polyps, right? But I was just wondering, because when you have a sustained inflammation, that can damage the body, right? And that's maybe why we don't know. And regarding this, I was another question I have is, can you have a allergy or more sensitization to cow meal's protein in a subclinical way? So you don't go to the hospital because you don't have symptoms, even though you might have some polyps, and then that can develop into some other type of inflammatory disease in the gut?
Yes, I believe this is, I believe it's more common than we think, because this case usually bleeds because the polyp moves within the rectum or the colon, but some of them have very small polyps. So maybe many go by undetected, and the child or the adult has just a rhinitis, or a chronic sinusitis. my son is allergic to cow milk allergy, and what he has is only a chronic sinusitis. He loves milk, so he can't stop drinking it. It's a bad example. He's old now. I mean, he's an adult. He can do what he wants.
Then he just leads with rhinitis, right? With
it's part of his life.
Exactly.
He does. But it's probably due to that because he's allergic to cow milk.
So where do we go from there, Cecilia? After this, of course, this type of work may be very important for T-cell targeted treatments, right? But what are the next steps? What do you think is crucial to investigate?
Well, we're actually we're studying different aspects of these polyps. First of all, we would like to know how microbiota influences the epithelial layer. In order to, we probably think we have alarmins like alpha-3 that are inducing this Th2 response. So we're studying that with epithelial organoids from the polyps. Then we're very interested in the mechanism that's generating these polyps and this allergy in patients mostly. So levels of Th2 cytokines secreted by the T lymphocytes were rather low. So we think probably when we compare with the values of Th2 cytokines we have in polyps, that probably we have LC2 producing a great amount of cytokines. So we want to find these cells. We're searching for these cells. It's not easy. So that's, and then the other aspect we're most interested in is in deciphering how more about IgE synthesis. I mean, whether this, if there we have a high affinity IgE, mostly because these kids don't have anaphylaxis, so maybe we don't. And mostly that mechanism, and we're searching in polyps for IgE memory cells in order to, we're more like in the study of the mechanism of food allergy. Of course, I would like to study further the mostly whether these T cells we find in polyps, whether they're T resident, we'd study T69 and other T resident cell markers, but mostly from that point of view.
Excellent. Well, we'll leave you to it, Cecilia. As I was mentioning, it's still a lot to be done to understand those juvenile polyps in allergy. Thank you so much for being with us on the podcast, and we look forward to reading your future manuscript.
Thank you very much for inviting me. It was a great pleasure, a great honor.
Thanks for tuning in to the Mucosal Immunology Podcast. The Mucosal Immunology Podcast is produced and brought to you by the Mucosal Immunology Journal and the Society for Mucosal Immunology. Thank you to our content contributors, Drs. Marian Brunk, Agnes Garcias-Lopez, and Ashu Mangalam. This podcast is produced by Matt Close and Catherine Canoop. We hope you enjoyed this episode. Remember, the journey of discovery in mucosal immunology doesn't end here. Stay connected with us for more in-depth discussions, expert interviews, and the latest research breakthrough. If you found this episode valuable, please subscribe, rate, and leave us a review on your favorite podcast platform. Your feedback helps us reach more researchers passionate about mucosal immunology. If there are questions or topics you'd like us to cover in future episodes, we'd love to hear from you. Connect with us on Blue Sky, LinkedIn, Facebook, or Twitter/X. Thank you for listening.